Hello, Assalamualaikum. Not sure how this is gonna work or how this is gonna sound, but it's very impromptu. Everything's very impromptu these days. But let's just get through this. So, let's close these. So yeah, to start off, mm, 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 mm. Sorry, this is a big mess. That's just how it's gonna be. So we're going to start off with learning about the control of internal environment. Now, in the last topic, 7b, we're not going to do too much of a recap, but near the 7b, we did learn about how for the um, essentially the control of the for breathing respiration to occur, as well as to increase the heart rate, to increase the cardiac activity, we use hormones. Okay? Whether that be adrenaline or electric impulses, but hormones are used or can be used, and it's going to be secreted through the medulla. Yeah, that is the center that we were learning that all these are going to be secreted from noradrenaline and adrenaline. So um, now we're learning more about what the hormones actually are. Generally, if not just from the medulla, where else can they be secreted from, etc. So let's go. So essentially, we start off with learning about with a simple definition of what hormones are. They are organic chemicals which are produced and secreted into the bloodstream by endocrine glands. So endocrine glands are going to be the things which always produce and secrete these chemical messengers or chemical sorry, not these organic chemical messengers called hormones. They are going to go towards target organs, and they're going to cause changes there. So, kind of like a transcription factor. Maybe can you argue that our transcription factors? Transcription factors are not hormones. Transcription factors are proteins that bind to DNA to turn genes on or off. While hormones are chemical messengers that circulate through the body, some hormones like steroids work by binding and activating specific transcription factors to regulate gene expression. Transcription factors, these are intercellular. Well, we're going to be learning more about hormones right now, but transcription factors are proteins which are intracellular proteins that directly interact with DNA to regulate gene expression. Hormones, on the other hand, are chemical signaling molecules that travel throughout the body to send messages. What these hormones do is that they pass into the cell and then they bind into these receptors, which are a type of transcription factor, triggering them to initiate a gene transcription. So they essentially are the things which trigger those transcription factors to start working on their cell, on the cell DNA. So the transcription factors then activate or repress gene transcription, acting as the final step in many signaling pathways, including those initiated by hormones. So yeah, can we just whoa, cool, interesting. All right, so essentially hormones and transcription factors work in tandem. You can't have them working separately. Okay? Unless the hormone is the ones which enter the cell. Pretty sure that's the other one, the one that is not a steroid. But let's keep reading. So, cells of target organs have specific receptors on their membrane and also inside the cells that are complementary to specific hormones. So, as with any protein, whenever we have things which bind onto something, um, we need to have specific receptors. The important part is that we have them in the membrane of the cell, of the target organ, as well as inside the cell of the target organ. Okay, and of course these are specific, so they're going to be complementary to something. What are they going to be complementary to? To the specific hormone. So we have two types of hormones: peptide and steroid. Steroid is what we've already seen already, that they're going to be binding to a hormone receptor inside the cell, which then binds uh, Okay, both of them involve transcription factors, but okay, here we go. So peptide hormones are made of amino acids and are water soluble but lipid insoluble. So remember, if we can't pass through it, that means it has to be lipid insoluble. As in, we cannot pass the phospholipid bilayer. So if we can't pass through the phospholipid bilayer, that means you know for sure that it's going to be binding to the membrane receptors. After which, it's going to stimulate the production of a secondary messenger, which is going to be acting as a transcription factor, regulating gene expression. Okay. Steroid hormones, on the other hand, are lipid soluble, so they can pass through the cell membrane, and so then when they bind with specific steroid hormone receptors, which are present inside the cells, they're going to be forming this complex, the steroid receptor complex, which then passes to the nuclear pore and acts in of itself as a DNA transcription factor to regulate gene expression. Here we have some examples as well, where peptide hormones, that is a mosquito, you annoying little thing. Okay, so we have some examples too, where the hormones for the peptide hormones are the common ones that we've learned already: ADH, insulin, glucagon, and adrenaline. These are the very common names that you see. Whereas the steroid hormone, these are going to be acting as more of the, how do I say, the more natural. For me, they were always this kind of more like intrinsic, natural hormone that gets talked about less in lower grades. This is going to be all estrogen, progesterone, and testosterone. These are going to be the sex hormones, if I'm not wrong. So note that the sex hormones and their derivatives, I believe, I guess, they are going to be the steroid hormones, whereas the things that are used for generally controlling stuff like water content in the blood, sugar content in the blood, heart rate, or you can argue cardiac activity. These are going to be involved, or these are going to be, um, controlled by transcription factors, which are going to be triggered by hormones, peptide hormones. Okay, so pretty simple difference between the two, right? Um, Now the specific glands, because we said that they get produced and excreted, secreted. They get produced and secreted by specific glands called endocrine glands, right? So what are these endocrine glands, or what can we know more about them? Um, how are we doing? Are we doing okay? I hope we're doing okay, because I'm really not sure. I guess we could do like this, right? This is probably better. It makes it a bit less scuffed. Do I have like a thing on? Wait, let me see if I can. Actually, I'm not gonna do anything. Whatever. Okay, so yeah. So let's learn more about these endocrine glands. We have not reached the second page yet. Okay. Okay. Endocrine glands secrete hormones, while exocrine glands release enzymes. So this is actually some extra information that we're learning. The endocrine glands are the ones that produce and secrete hormones, whereas exocrine glands release enzymes. So why are we learning about exocrine glands? Well, endocrine glands are ductless and directly secrete into the bloodstream, while exocrine glands have duct and don't secrete directly. So completely the opposite. So how, steroids and peptide hormones are kind of the opposite. Endocrine and exocrine are completely the opposite. Endocrine glands are ductless and directly secrete. Exocrine glands have a duct and don't secrete directly. Let's just look at a picture of that. So you can see endocrine is ductless and is directly secreted into the blood, whereas exocrine has a duct and doesn't secrete directly. So this is going to be your duct. I'm not really sure exactly how that works, but anyways. So moving on. Endocrine glands include, because we're learning about endocrine only, right? The pituitary gland, which is going to be the master gland. The hypothalamus is going to be used for thermoregulation and osmoregulation. So you can immediately tell for temperature and water content. Thyroid glands, pretty sure this is like in the throat region, involved in controlling growth and metabolism. I don't think I'm supposed to be remembering these because how the hell would I remember these? But it's good to just read through them. So pituitary, hypothalamus, thyroid. Then we have parathyroid, which is going to be involved in the homeostatic control of calcium metabolism. Pancreas, adrenal glands. So remember the adrenal glands. This I think got mentioned once. So of course they're going to be using for adrenaline, aldosterone, kidneys, ovaries, and testes. Ovaries and testes are going to be using for the male and female sex hormones, which are obviously going to be those peptide hormones. Those steroid hormones we were talking about earlier. And the kidneys produce hormones involved in the production of red blood cells and vitamin D metabolism. Red blood cells and vitamin D metabolism. So we do know that the kidneys contain. Actually, I'm not sure, but okay, but I guess just keep that in mind. Okay, nice. So we know that the pituitary gland is the master gland, but it is controlled by the but it is controlled by the hypothalamus. The control of the pituitary in a hormone, uh, the control of the pituitary in the hormone release and homeostasis. So, oh, I did write some stuff here. Cool. So, the hypothalamus lies. Okay, so when we look at how the hypothalamus and the uh, oh my god, that was a mosquito. Holy shit, that's crazy. When we look at how the hypothalamus and the pituitary gland work together in hormone release and homeostasis, note that we're also talking about homeostasis here, okay? Because we're trying, because that is the overarching function of these, you know, hormone release, where it's like, well, not always, right? But like if we're looking at, for example, the peptide hormones that we were looking at, their function was maintaining an internal environment. Maintaining an internal environment is by definition the function of homeostasis. So that's only going to be possible, or that is going to be achieved by using your hormones, which are going to be secreted by your pituitary glands, which work in tandem with your hypothalamus. So the hypothalamus lies above the pituitary and controls its activities. The interaction between hypothalamus and pituitary control homeostasis. So we're going to be having a relationship with the hypothalamus and pituitary, and that's going to be controlling homeostasis, or it's going to be controlling the hormone release, by extension homeostasis. Okay, hypothalamus controls pituitary activity using neurosecretory cells. Let's call them NS cells. Okay, NS cells, which are cells which produce secretions from the axon. Now there are two types of such cells: NS cell 1, NS cell 2. NS cell 1 produces substances releasing or release which are releasing or release inhibiting factors that stimulate or inhibit hormone release from the anterior pituitary. So axons from the hypothalamus produce secretions which uh, which are of two types. Okay, so uh, neurosecret neuro, neurosecretory cells are two types. Essentially, they have an axon from which secretions are produced. Now, in NSL1, these secretions are going to be release inhibiting or releasing factors which can cause the amount which can stimulate or inhibit the hormone production and release from the anterior meaning the front anterior pituitary. Okay, and then the posterior pituitary, let's say near secretory cells 2 produce secretions that are stored in posterior pituitary, which are released uh, as hormones. So we have NSL2 secretions making hormones in hypothalamus, release synthesis plus release, stored until impulse causes release. So essentially, near secretory cell 2 just causes secretions that are stored. Essentially, you can imagine that it makes these hormones which are just staying in the posterior pituitary, and then depending on whether the NSL1 or the anterior pituitary uh, stimulates the release of those cells, that is effective. Okay, so there you go. So yeah, I don't think this is super important, but NSL1, NSL2, NSL1 works with the anterior, NSL2 works with the posterior. This one kind of does the synthesis plus release. This one does kind of just the storage. Okay. There's a close structural relationship, and remember the hypothalamus is obviously on top. Remember that the close structural relationship between the hypothalamus and pituitary is reflected in their function. The hypothalamus produces near secretions that control both lobes of the pituitary gland. Okay. Action of peptide hormones and steroid hormones. We already know overarching how they differ, but if we look into the specific mechanism, I guess you could say. So we already have the names: insulin, glucagon, ADH, adrenaline, noradrenaline. Using ADH as an example, so we're going to be not only just looking at how it works, but its cause and effect, right? So using ADH as an example, when you have ADH or the peptide hormone binding to the specific receptor on the cell surface membrane, this stimulates some chemical reactions in the membrane, resulting in the production of an enzyme. So cell surface membrane receptor binds to that, chemical reactions producing an enzyme. Enzyme and ATP result in the production of a second messenger, commonly CAMP. Then the second messenger moves to the nucleus and acts as a transcription factor switching on or off genes. So essentially, it doesn't directly make the CAMP or the second messenger, it makes the enzyme or the production of an enzyme, rather like when it binds, that causes reactions, which really results in the production of an enzyme which binds with ATP to then form the secondary messenger, which is CAMP, which acts as a transcription factor. So it'll enter the nucleus to the nuclear pore, and it'll start just messing around with the DNA, acting as a transcription factor. Now, steroid hormone, on the other hand, is going to be liposoluble, so it's going to be entering through the cell membrane and acting as an internal messenger. As it binds with the steroid complex inside the cytoplasm, specific and complementary to it, it's going to form a steroid receptor complex, and it's going to be passing through the nuclear pore as the CAMP does as well. And it's going to be acting as a transcription factor, switching the genes on or off the same exact way. Protein metabolism in the liver. So this is that was the whole one thing that we were learning. Now we're looking at examples of essentially um, homeostasis examples, kind of, I think. So okay. Okay, so protein metabolism in the liver. So this is pretty cool because we're learning about how proteins are made, as well as, or is it about how proteins are made, or is it how, I'm not sure what metabolism, what does metabolism mean again? What is the definition of metabolism? Metabolism is the continuous 24/7 set of chemical reactions in the body that converts foods and drink into energy. So how we use how we use proteins as a way to generate energy. Keto acid. Okay, we'll see. So liver cells, hepatocytes, remove the amino group. Okay, so each of these come as 3 or 4 marks, and we're learning what the metabolism process is by the liver, by the yeah of the proteins. So liver cells, which are hepatocytes, remove the amino group or deaminate, because deaminate amino acids, converting it into ammonia. So we're trying to remove this amino group from an amino acid. Okay, we're going to be left with ammonia, and we're going to be left with this kind of chemical or compound called a keto acid. Okay, so this keto acid is going to therefore be used. Uh, okay, wait, hold on. So then the ammonia is going to be combining with carbon dioxide in the ornithine cycle. The ornithine cycle then produces urea, which is less toxic than ammonia, and is going to be excreted out. Remember when we mentioned deamination is going to be used? Well, deamination was not actually the process that produced urea. Deamination is a process of turning amino acids into breaking that down into just, or actually just removing the ammonia from amino acids and then using that as a way to turn that into urea, which is less toxic than ammonia. Okay, so that's going to be excreted out. The remainder of the amino acids, which is going to be the keto acid, is going to be used as in the respiration in the Krebs cycle, are going to be converted into lipids for storage. Contributes to the Krebs cycle one of the intermediates, alpha ketoglutamate. I'm not sure. I think this is just a guess, though. So you can see over here that ammonia goes in, and water goes out, water in, urea out, carbon dioxide in, water out, then ammonia in. So you say carbon dioxide is going to be in, it's going to be binding with the carbon dioxide of some sort. So it's going to be binding with the carbon dioxide in the ornithine cycle, and at the end, urea is going to essentially just be popped out. Okay. Details of the cycle is not given, as not required. Given info above is from MS. No more detail needed than that. So again, liver cells hepatocytes remove the amine group from the amino acid, convert it to forming um, ammonia. Ammonia is then fed into the ornithine cycle where it combines with carbon dioxide. Uh, Urea is produced, which is more stable or less toxic than ammonia, and it's going to be excreted out. And then the rest of the amino acid, which is a keto acid, it's going to be it is going to be used in the Krebs cycle or used as a lipid, converted to a lipid for storage. Okay, third page. So I think if we get through this third page, it'll be pretty good because there are quite a lot of information. So these last 2 pages are crazy. This page is probably the actually no, well, to be fair, this page is probably the hardest. Anyways, So that is how we control, that's how we control the, essentially, not really controlling anything, but we're able to turn amino acids into something a lot more stable. We're able to turn them into something that's going to be used in the Krebs cycle for releasing energy. We're going to be able to convert them into, remove the most, toxic part of the amino acid and convert that, and then excrete it, convert it into something less toxic, and then excrete it. So overall, it's good. This is all going to be done in the liver. Okay, so there you go. Now, urine formation and osmoregulation are done by the kidneys. Now, urine regulation and osmoregulation should be hand in hand. The kidneys also do homeostatic control of water potential, okay, urea and toxin removal from the liver, right? This stuff which came from the liver. Now, urine formation has three main stages. Total mechanism given below. Ultrafiltration. High pressure, hydrostatic pressure due to large diameter. Okay, let's watch a video on this because it's gonna, urine formation process. Yeah.
The kidneys filter blood
Holy
plasma,
shit!
removing metabolic wastes, toxins, and excrete
The
them in
buttocks.
urine. During this process, they also maintain constant volume and composition of the blood, or homeostasis.
See, we're just
Blood
seeing that shit.
enters the kidney.
The comments gonna be talking about it. In no fucking way. Okay, never mind. I'm just. I'm sorry. Uh.
Kidney via the renal them in urine. The kidneys filter blood plasma, removing metabolic wastes, toxins, and excrete them in urine.
Yep.
During this process, they also maintain constant volume and composition of the blood, or homeostasis.
Yep.
Blood enters the kidney via the renal artery, which divides into smaller arteries, then arterioles.
Yes.
The arterioles get into contact with functional units of the kidney called the nephrons.
Okay.
This is where blood filtration and urine formation take place.
Okay.
The filtered blood then passes through a series of veins and exits the kidney via the renal vein.
Okay.
The urine is collected in collecting ducts and leaves the kidney via the ureters.
The filtered blood leaves through the renal vein back towards the heart, and the urine is collected in the collecting ducts.
Urine is
and then leaves
collected in collecting ducts and leaves the kidney via the ureters. Each kidney contains over 1 million
So this is the filtering thing. Okay, the filtering thing, and plus where the urine is collected.
nephrons.
All of this are going to be sent to the collecting duct, which is going to be then is going to be then excreted via the ureter.
Nephron consists of two major parts: a glomerular capsule, or Bowman's capsule, and a long renal tubule. Renal tubules of several nephrons connect to a common collecting duct. There are 3 steps in the formation of urine: glomerular filtration, tubular reabsorption and secretion, and water conservation.
This is the same thing that we learned here. Ultrafiltration, glomerular filtration, selective reabsorption. What do you say here? Tubular reabsorption and secretion, and water conservation, and they mention tubular secretion, selective reabsorption, tubular reabsorption, tubular secretion, tubular secretion. Okay, I'm not sure where that's going to be, but okay, let's see.
Blood enters the Bowman's capsule via the afferent arteriole,
So
passes through a
remember,
ball of
this is
capillary.
this is the thing that came from the renal artery that divided into the arteries, um, that divided into the arterioles and such. So now this is gonna be still containing the original blood, right? Just in a much smaller sized artery that's higher surface area, etc.
Capillaries called the glomerulus, then leaves via the efferent arteriole. The afferent arteriole is significantly larger than the efferent arteriole, creating a blood flow with a large inlet and small outlet. As a result, the blood hydrostatic pressure in these capillaries is much higher than usual. Hydrostatic and osmotic pressures drive water and solutes from blood plasma through a filtration membrane into the capsular space of the nephron. The filtration membrane acts like a sieve, allowing only small molecules to pass through. These include water, inorganic ions, glucose, amino acids, and various metabolic wastes such as urea and creatine. Glucose, amino acids, and various water into the capsular space of the nephron. The filtration membrane acts like a sieve, allowing only small molecules to pass through. These include water, inorganic ions, glucose, amino acids, and various metabolic wastes such as urea and glucose, amino acids, and various metabolic wastes such as urea and creatinine, and make up the glomerular filtrate. The amount of filtrate produced per minute is called glomerular filtration rate, GFR. GFR is kept at a stable value by several feedback mechanisms within the kidneys, known as renal autoregulation. GFR is also under sympathetic and hormonal control. GFR regulation is generally achieved by constriction or dilation of the afferent arteriole, which causes the glomerular blood pressure to fall or rise, respectively. In a healthy person, the total filtrate volume amounts to between 150 and 180 liters a day. However, only about 1% of this is excreted as urine. The remaining 99% is reabsorbed back to the blood as the filtrate flows through the long.
Okay, so from what I understood, right? So we have the afferent and the afferent arteriole, right? So when it does enter, okay, uh, yeah. So when it enters through the blood, when it enters into this kind of, I always think that the door is opening. I don't know if it's, is it a cockroach on the fucking, on the couch? Like, are we for real? It is. How, how the hell did you get on the couch? What are you talking about? Okay. So, yeah, okay, maybe tomorrow. I'm not dealing with this bugs. Sorry.